get started. So hello, everyone. My name is Casey. I'm from Cambridge University Press, and I would like to welcome you all to today's session of Digging Deep with Quantitative Plant Biology, the webinar series. For those of you who are new or who don't know, the Quantitative Plant Biology is an open access journal published by Cambridge University Press in association with the John Innes Center. We're delighted to be bringing you this webinar series thanks to our editors, authors, and the support of the John Innes Center. In a moment, I'll be handing over to our editor-in-chief, Olivier Hamont, to introduce today's session. But before we get started, I just want to do a few housekeeping items just so you know how to participate in today's event. So you have all joined in a listen-only mode, which means you'll be able to hear us but not able to speak to us. However, you will have the opportunity to submit chat questions. So down towards, usually to the bottom of the screen, sometimes at the top, there's a chat function as well as a Q&A function. You can use either of those to submit any questions at any time during today's webinar. And we'll collect these, and at the end of today's session, we'll have a little Q&A session as well. If we are unable to get through all of those questions, we'll also have a follow-up email where you'll be able to submit questions that you think of in post or any that we don't get to today. Should you experience any technical difficulties today, just let us know in that chat box as well, and a member of our team will help you get sorted there. And in the unlikely event we do experience any issues, we will message the audience and restart the webinar, but that shouldn't happen, hopefully. So to summarize, you can submit your questions at any time, and you'll receive a follow-up email to let us know how useful you found this and have another opportunity to ask any questions. So to kick things off, I will hand over to the editor-in-chief of Quantitative Plant Biology, Olivier Hamont.
Thank you, Casey. Very happy to be here. So maybe just a few words about Quantitative Plant Biology. So it's a journal that is community-based. And so for a community-based journal, we are very fond of tools that we can share with the community to help the community get into systems biology, better understanding of their model systems, and to ask new questions. And so this is exactly what we have today with the Leaf Quantitative Atlas. So I'm very happy to have Dimitri and Gilles here today. So well, maybe I won't talk about the Atlas. You're going to talk about it, but I'm very happy. I'm going to leave you the screen. So thank you. Thank you for being here.
Okay, shall I share the screen?
You can share the screen, yeah.
Is that okay? No, it's still.
So it's not full screen yet, but yeah.
Yeah, it's okay. So go back a little bit. So thank you very much. Hello, everybody. Thank you very much for the organization and for the kind invitation to present this, let's call it tentative, Arabidopsis leaf quantitative atlas. So this is a long story that started maybe 10 years ago. It's rooted in an experience in modeling of metabolic enzyme systems for my part, and on cell biology, plant cell physiology, and anatomy for Dimitri. So it's rooted also in the conviction that quantitative analysis is a good way to really understand how biological systems work. It's also a way sometimes to reconcile apparently conflictual results, and it's also a way to simplify. I will not substantiate further, but that's something that is maybe paradoxical, but that it's when you tackle things quantitatively, you can simplify. So I will try to go to the next slide, if I can. Yeah. So the talk structure, we will start with more details on the motivations, the method we used, the atlas content, and the organization of the data. So it will be kind of a tutorial, a way to present data that is different from the paper and maybe more convenient for you. I will follow with the use case, some words on data accessibility, beside what is available in the paper, in the manuscript. And I will finish with highlights of some potential interesting quantitative data. You know, it's a kind of things, the same as when you are in a dark room and you develop your photo, what you have at the end, and it's always interesting when you have integrated all the information. Okay, so very practically, this is probably a question that you had if you're a biochemist or cell biologist. You will have to do an experiment. You would like to be in the so-called physiologically relevant range, and you should say, how much of this metabolite should I put in my tube, or when you write your protocol in your lab book. So you will go for Google. In 0.3 seconds, you will have 3,000 results, and then you will spend hours to read and find interesting value. If you're lucky, you will find the value, the concentrations in micromolar directly or millimolar, directly useful for your experiments. But most of the case, you will find these baroque units per square meter, per gram fresh weight, per gram dry weight, and then you will have to search for additional information, water content, cell volumes, organelle volumes, if the metabolite you're interested in is only in one organelle, cellular localization, etc. And then you will have to do calculations, and then finally you will have the result. So this could be discouraging, it's time consuming, it's repetitive, sometimes it's uncertain. You're never sure that the value you have is actually corresponds to the state of the plant you're working with. In a different context, the context of metabolic modeling, we already have tools to build metabolic networks. We have either the models that people built previously, you can rely on this, you can build on this. We have a lot of knowledge base or database, including ours, that has a specificity for KB. If you're interested in molecular regulation, protein association, subcellular localization. But when it comes to define initial conditions, if you do kinetic models or boundary conditions for steady state models, it's much more tricky. You need a huge amount of information on the physiological context, what is around the system you try to model. You can rely on bionumbers, but bionumbers is limited in the amount of information it's present, and you will also rely on literature. And all of this is generally not sufficient. It's time consuming, and it also could be discouraging. So this is the reason why the real motivation based on our experience in dynamic modeling to provide a generic tool for the modeling community, to avoid that people would have to do all the time the same things because it could be done once for all. So some methodological points. It's very basic, but I think really important. To build an atlas, we need a reference plant developmental stage. It might be quite limited first, but we start to do pictures because before doing movies. And it's considering the large amount of data you need, you can first start with a given reference developmental stage. And you also need a reference growth condition, something that is well measured, well quantified, generally in phenotyping platforms. And this will consist, let's call it islands of stability. This is something that is a concept that is interesting in science. It's something that is maybe not perfect, maybe not finished, it's not definitive, but it's something kind of a focal point on which we can meet around, discuss, maybe disagree. There's no problem with that, but it's something that, well, it's kind of a reference. This is what an atlas is for me. So in terms of method, if we go a little bit deeper in the details, the reference state we choose, the developmental state we choose, is the 1.09 defined by Boyce in 2001. You will see why in some slides, why we choose this reference state. Leaf 6, because it's the first leaf that is mature in Arabidopsis, and many of the data were collected on leaf 6. There's something else that I think is interesting that we discovered during the building of this atlas. It's the leaf developmental stage that integrates the leaf surface at time t of the measurement, or when you take the leaf to do your experiment, divided by the maximum leaf surface, and you add the number of days after maximum extension is reached. And it's something that apparently is not very used, but that could be useful because there is, if you're interested, you can read this paragraph in this paper about controversy in the leaf developmental stage. So in our case, the leaf we will use for the atlas, it's not completely expanded because the experiments were done with this kind of leaf. So it's the LDS is 0.05 84. Okay, the reference growth condition, it's based on data that were acquired a long time ago, 10 years ago, in the Montpellier phenotyping platform in France, in the laboratory labs. So this corresponds to long day conditions. You all know that plant phenotypic plasticity is quite large, so we need to be sure of, at least to clearly define the growth conditions we use. So the plants for this atlas, you see here the growth curves of the plants. The top curve is the well-watered plants. The leaf that was used, we see this just after, is under this condition, 21 days, a surface of 1.2 square centimeter. And with that leaf, Natalie Witz did a very nice work. At that time, it was really pioneering using multiphoton scanning microscopy. And we'll just need to move a little bit this so that I can see my screen. So she took a small part of this leaf in the middle of the leaf, supposed to be representative of the rest of the leaf, except for veins. We will see this in a while. So in terms of method, somehow the method was determined by the available information. So we selected this work because it was the most quantitative source of quantitative anatomical data for Arabidopsis leaf. So somehow this also determined the growth condition we choose. So we had this kind of binary, let's say binary way to analyze data. If the data were available, we will integrate it in the table S1. So table S1 is the atlas. It's the core of the work. If data were not available, for example, in that study, it was not available for vein cells, we had to search in other studies. And again, yes, no. And sometimes we had to degrade our standards because we could only find data in different growth conditions. So this is coded. We'll see this in a minute. And if data are not there, well, that's indicated missing data. And it's also an important point when we assemble the test to know what we know and what we don't know. Recently in a seminar, a model said that you can't imagine how many things biologists don't know. And it's a way to illustrate this. Okay, for the subcellular matrix of the leaf cells, not surprisingly, the amount of information is lower. And very quickly, we have to degrade our standards and use different growth conditions and even different plants. So this might surprise you, but if you're interested in chloroplast, for example, you have to rely on old data that were acquired on spinach. And this is also a way to show that there is some room for progression, improvement. So everything, the idea of the atlas is to have to be quite transparent. So in the Excel table, you will see some cells that are boxed or surrounded by colors. And this corresponds to this code. If it's orange, it means that the information we have comes from different growth conditions. If it's in blue, it's extrapolation. If it's in violet, it's low quality data. For example, when there's only one measure. And if it's in green, it's because it's not from Arabidopsis. Finally, when everything was integrated, we could do some calculations, and we obtained this table S1 that is actually 10 tables. We will describe this later. From this table, we extracted the two tables that are in the paper, table 1 and 2. And we also have a use case. From this information, we could derive conversion factors to obtain a common unit for the different data we could find in the literature. This is table S5. So an overview of the atlas content. Witz et al. Study, Natalie Witz et al. Study produced information for 4 kinds of cells: the epidermal cells and the palisadic and spongy mesophyll cells. The other cells and the data were not available. So for example, for bundle sheath cells that you see in light green on the left, or the xylem cells or the phloem cells, this had to be extracted from other studies, and sometimes for different growth conditions. So now we enter into the hard stuff, this table S1 that you can't see at a single glance when you look at it in the computer. So I placed it horizontally, and I will try to help you and guide you in this table. So if we start with high-level information, you have on the top the leaf metrics, below the cell metrics, then the organelle metrics, chlorophyll units, and conversion factors. I will detail that later. So if we go into more detail, you have the leaf, cell density, cell numbers, individual cell volume, total cell volume per leaf, information on the sap, apoplast, airspace volume, etc., and different units. For the organelles, you have all the organelles of the leaf, of the cell, in all kind of cells. So you have color codes for the different cells, and the legend, you will find it in the table S110. Okay, we zoom on this table. So on the left, you have the headings, then you have the variables, the values, experimental error when available in the papers, min-max values, units, references, comments, for example, explanations and calculations, references to figure in publication. So that's very quick. If you read the comment and you see this is figure 6D in this paper, you can check and double check. And it's also a way to see if we make mistakes or not. The method, because the experimental method determines the quality also of the data. Growth conditions, they are similar or different, and plant stage. A very important visual code. If you look, you will see that some figures are in regular, and others in bold. So when they are in regular font, that means that they are from the paper published. Sometimes with a precision that is maybe illusory because of we know that experimental measurements in plant is subject to variation. But we prefer kept this illusory precision because it's easier to check and to look at the value in the paper directly. And when it's bold, it results from calculation. So you can check on the cell and you will see, for example, here, you will see that the cell D14, you will see the equation. So it's a way to check how we did the calculation. Sometimes it's a little bit more complicated because we had to do calculation using data from different tables. So we might in the future think of something more easy to display and to follow. Okay. So you probably have seen that there is not a single table. It's not the table S11 that contains all the data, but there are also other tables, especially for veins, because we need special calculations. So you will learn in this table that, for example, this small leaf of 1.2 square centimeter contains 43 centimeters of veins. And here it was quite frustrating because to do these calculations of cell numbers, etc., we need the number of cells per minor vein cross-section. And actually, this information is not easily accessible. So we have to rely on different studies. You will maybe see in this extract of the table, you see there's lots of orange. This means that it's not under, they were not collected under the reference growth conditions. But still, we need numbers because if we want to do calculation, we still numbers. So we know it's not satisfying. We need to improve that. But it's used to feed the other table and obtain values. For ER and Golgi, it's even worse because lots of people do some dynamic analysis of ER, but you could imagine that from the shape of this organelle, it's very difficult to calculate volumes, and we have to rely on very old 2D analysis from the old days. And even in different plants, you see the green in the table, and the violet and the blue. That means that there's lots of hypotheses. So here again, some room for improvement. Finally, a table on the membrane surface and cell wall volume. We will detail that in a while. So here, maybe I will not just detail. You will have the video afterwards on YouTube, so you can screen capture if you want to, if you're interested in a specific matter, to see how it is organized. table S18. It's about subcellular data. So we simply synthesized what was calculated in different tables. We grouped everything in a single table where you have the different organelles and different volumes of these organelles depending on the reference you want. So surface, fresh weight, chlorophyll, percentage of leaf volume, etc. And from that, if you need something else, you can do it yourself, the calculation. Some more details on how we model cell surfaces, excluding the cell wall. We see cell wall afterwards. So for spongy mesophyll cells, we simply model them as spheres. We know the volume from Vuit's et al. Study, so we can deduce the radius and then the surface. For parenchyma mesophyll cell, it's a little bit more complicated. We have the volume. We can, it's a capsule. So the surface is more complicated to calculate, but you have here the procedure. So this is hidden in the table. This is the visual representation of what we did. For bundle shift cells, it's a cylinder. For vein cells, also cylinders. Gourd cells, a tor. And finally, epidermal cells. It's quite complicated to have the surface of a puzzle piece. I have a supplementary slide to describe that, but I think it's not the point here. If you're interested, I can provide you the information. The idea is that it's not trivial. Okay, so a use case when you have assembled all of these data, you can calculate conversion factors. So you will find them at the end of table S11, the lines 943 to 1000. So the idea is that if a metabolite is in all cells, all subcellular compartments, you can calculate a conversion factor to convert units that are nanomole per gram fresh weight into micromolar. So you see it's close to 1. So if you have 1 nanomole per gram fresh weight, you will have 1 micromolar. But you can have also other possibilities. If the metabolite is in the vacuole, the chloroplast, the mitochondria, or in only the chloroplast, you will have different conversion factors that are here. We make an assumption that the concentration is the same in the different compartments. We can't do better than that. We have lots of quantitative data that have been collated for maybe 10 years in ChronoKB. So it's not actually 640 metabolites, it's 640 data on metabolites. Sometimes we have many data for the same metabolite. We have the various cell and subcellular localization, various experimental methods. So this is table S3 that you will find in the supplementary material. It's what we can export directly from ChronoKB. And then with a Python script, we were able to convert all these data in the variety of the units in a common unit in micromolar. And this could be used as constraints for modeling. This actually had been used by one of the co-author, Ed Smith, in thermodynamic modeling. And actually, it was a way to have his model converging. So somehow it appears to be useful. Just a small description of table S5. You know that the metabolite name is quite variable, and it's sometimes difficult to, from one model to another model, the name is not the same. So we introduce in this table all the other way to name or to characterize these metabolites using KEGG, Metasci, CASP, INCI, SMILES, etc. Codes, lipid maps that is not complete for plant lipids. And you have the quantities in the middle, the units, and the source biological material. For example, if people started from roots or started from leaf or started from isolated chloroplasts or thylakoids, you have information on where it was quantified. If it was quantified in, I don't know, certain tissue or certain subcellular compartment. And finally, you have a correlated localization. So this is something that's introduced manually in ChronoKB. For example, it says that this metabolite, let's say phosphoglycolate, it's only in the stroma. You have the species comments that integrate the conditions, the growth conditions in the study. Then the concentration in nanomole per gram leaf fresh rate, that could be useful for some people in modeling rather than directly micromolar that we find at the end. And we repeated the name and the localization so that if you search one by one these metabolites, it's easier. So the part in green is common with table S3, and the Python script was used to do the final conversion. Okay, so for the data accessibility, we placed it in addition to the quantitative plant biology website. You have them in Zenodo. You have the Python script, table S3, table S1, which is the atlas, table S5, and this is also a way to publish updates. When there will be version 2, version 3, depending on new data we will analyze, and at each time there will be a new DOI. You can also, you see the connection with CuroKB. There's a download icon on the top on the welcome web page, and you can go there to the downloadable documents, and the table S3 is actually available also here, and it's frequently updated. Okay, I will finish with highlights on some potential interesting quantitative data. So this is the front part of the atlas. So in terms of number of cells, when we looked at literature, actually, it's not easy to know how many cells you have in a leaf. Generally, you have the number of leaves per surface, for example, for the epidermis, but in an entire leaf, you don't have. And the surprise was to find that vein cells were nearly as abundant as photosynthetic cells. And this is something that, well, could make sense because they are very small, and they have functionally they are really important. Stomata guard cells also, it's quite generally when we see pictures on the web, we don't see the epidermis, the upper epidermis with stomata cells. Well, still there are some, and they are quite abundant. Everything is synthesized here in table 2. So you have nearly 1 million cells in a small leaf of 1.2 square centimeter. And you have here the different units, depending on the reference you need. For the cell wall, which is, if you do a global model of a plant, that is an important place where material is stored. And we could not find data for photosynthetic cells, so we had to produce ourselves on the data, thanks to Ray Whiteman, who produces very nice images. And this could be introduced in the table. These are the green squares. And there were some technical difficulties for veins, so then we had to rely on other works, stem analysis, and it was unfortunately under different growth conditions. But still, we can do some calculation. That's something you might ask, okay, you integrate many different things, leaf surface, number of cells, surface of cells, cell wall thickness that you measured with a certain incertitude. You assemble everything, you obtain this value of 0.7 microliter per leaf. The volume of this leaf is about 24 microliter. And this represents 2.7% of leaf volume. But yes, okay, but how can we believe in that? So there's a different way we can, and you will find this in table S16, you can use macroscopic data, people that did some macroscopic measurements. And if it's not immediate, you still need a calculation, but finally you find 3%. So, okay, that's, as I said initially, it's an atlas is an island of stability. It's a reference. It's a point where we can compare. If we disagree, then we will need to find out why there's a disagreement. If it's similar, well, it's reassuring. Okay, if we look at the membrane surfaces in leaves, so this is data that are not published. I did this quite recently. You have this small leaf of 1.2 square centimeter, and you can calculate the different surface of membranes, thylakoids, envelope, etc. And you see that the thylakoids represent a huge amount of these membranes. It's a total of 1,200 square centimeter. So this is an A3 sheet, and there's a 1,000 factor of amplification. So if you're interested in exchanges, transport processes, this could be interesting information. And this is a first calculation. I tried to calculate the volume it would represent if the membrane is 4 nanometer thick. And this would represent quite a large part of the volume of the leaf, about 20%. So this is surprising. As Dimitri said, when we start to do calculations, sometimes we're surprised. Maybe this is wrong, maybe this is true, and this is something that can change the way we think about physiology. So I would like first to acknowledge the different people who participated directly in this work and provided some information, various information. Dimitri, who provided nice inputs, especially on cell wall and cell and leaf anatomy. Ray for the SEM analysis, Natalie Witz, who provided the images, our funders, and I would like to thank you for your attention. And I insist on the fact that we are really interested in feedback. If there are errors, don't hesitate to report errors, to suggest improvement, to provide new data, or to let us know if new data are produced that are not introduced in the atlas. And if something is not clear, please ask. And you can communicate using the email addresses at the bottom or even the small envelope in the ProKB website. That's it. Thank you.
Thank you very much, Gilles. This was extremely clear, very conducive to use the tool, actually.
Thanks.
So maybe to kickstart the discussion, I couldn't resist quoting Darcy Thompson, because what you're showing is exactly in that pattern. He's talking about mathematical beauty, and he's saying, the harmony of the world is made manifest in form and number. And so this is what is beyond this Excel files, right? And I like your surprises with the vein and I mean the same proportion in veins and the photosynthetic cells, the 3% volume for the cell wall, the membrane. I mean, all these surprises. This is part of the mathematical beauty that you extract from this atlas. Something else I want to say is, and this is a bit behind the scene, but the reviewers were very enthusiastic about the paper. So I also want to convey this in this seminar. And then something that I also want to raise and to highlight is the posture that you have to be towards transparency, I mean, open access, like sharing, and also to show what we don't know, which is almost as important as showing what we know. And all this is in the atlas, so really congratulations for all this work.
Thank you.
To start the discussion, I can start, but don't hesitate for the audience to ask questions. I have maybe a first one, which would be, so the idea of having an atlas, as you say, is to have a reference, sort of an average, I would say. But you also have the error, right? You have the experimental error, you have a standard deviation. So maybe a first question, which is probably a difficult one. What is the most variable parameter in a leaf? Or do you have an idea of the one that are the most stable, roughly? I mean, of course, I'm not asking you to go through the table, but from what you saw, what would be the most variable and the least variable?
So maybe I can show, I prepared a slide on that. This is something that was published by the group in Montpellier. They tried to compare Arabidopsis grown in, I think it was 12 different laboratories. And they tried to indicate all the conditions that was the same, and they grew different ecotypes and the same ecotypes in the different labs. And you see the variability. And if you look at the data, you see some cases it's clear why there are some variability. In other cases, it seems that people grew the plants under the same conditions. So you see that it's quite large. The average is, it's fine. They identify a core labs where the parameters were similar, but there are large variation that are linked to the quality of the lights, the quality of the lamps. So directly, you see the surface you can have a factor of 2. But it depends on what you need. I think in terms of model, generally what we need is simply a range. When you feed model, you don't need precise value. I think generally I take the comparison of the electrons in the orbitals of an atom. You don't need to know exactly where it is. You just need to have a shape. And this is what you need here. You said, okay, in the model is that 1 micromolar and 10 or 1,000 and 2,000. It's completely different for models. And then you need only to have this range, and then you can decrease the complexity of calculation. This is the reason why I say that quantitative biology is a way to simplify. Some things that would be very complicated to calculate. If you now provide real ranges, then it could be much simpler.
Yeah, yeah. Agreed. I see that there is a question from Olivier Martin. Hello, Olivier. What is the fraction occupied by the apoplast? So the fraction, because you mentioned the volume, but for you, is this the same as the fraction occupied by cell wall? Ah, okay. So
there is a problem of definition here. I think what I introduced, it's probably explained in the table. It's the aqueous volume in the apoplast. So in the table, I do not include the vein contents is not included as apoplast, but here there is an ambiguity. In some papers, they consider that the apoplast is somehow, if you centrifuge a leaf, what you recover from the veins, slow centrifugation, is the apoplast, the entire thing that is outside that can be extracted from the leaf. I think that in the table, this is what is outside the cell wall. How it was measured, this maybe should need some more. So I read that. Maybe it's not clear enough, and it has to be more clear. Thank you for the question.
Maybe I guess a sub-question on this is because here you're measuring cell volume with electronic microscopy, so you could also dry a little bit the cell wall, right? So you could maybe underestimate the volume, like if you did quick.
Yeah, it's true. The block we see of the leaf, it has been prepared, fixed, dehydrated, and well, we have new techniques now that use fast cryopreservation. That's a way to keep the volumes. And I think, well, the very last data we had are published very recently. It contains the veins. I'm really interested in this data, but still there were chemical fixation. It was not with these new methods, but the new methods might be a way to clarify. After that, when we compare confocal data with this data, we generally arrive at the same volume. In this paper, for example, the chloroplast, they published it in just at the same time, and the chloroplast volume was 90 microliter, which is exactly what we have here using FIPSEM, and they used confocal analysis. So I think that's also a way we compare method, and if it's fine, this is reassuring. And if there's a difference, we need to integrate it as a correcting factor.
Yeah. And I have another question. I mean, one of the surprise, or maybe it's not a surprise for you because you're working on this type of objects, but the fraction of organelles. So basically, the atlas is also showing that it's a bag of organelles, right, of thylakoid membrane and of organelles. And so, I mean, it could make sense for a leaf, right, because it's a photosynthetic organ. But I'm thinking of, I mean, I've been in a few review panels for projects in animal science, and organelles are growing a lot. I mean, as a subject of interest in animal development and cell biology, there's almost like a drive towards organoids and organelles, and so we sort of leave the cell a little bit out. And so I'm wondering with your atlas if you might contribute to this trend as well in plant science, that we so often forget that the unit of life is maybe not the cell exactly, but more at another level. So I don't know if, I mean, I'm not sure this is it's more a comment or a question, but I don't know what you think of this view on organelles, how it is evolving in time in plant science, I guess.
Well, I think it's, for people, it's important. In models, the first models considered the plant as a bag, but now it's all the models integrate the different organelles and the transport steps. It's really important. Well, maybe it's an anecdote, but some reaction in metabolism occur in the ER. And is it why? It has to go inside the ER. The ER is a very small volume, so it's a way to concentrate the metabolite and to increase the rate of reaction. So an enzyme that would not be very efficient if it's localized in this small compartment. So this, to me, it's really important in terms of isolating processes. the mitochondria and the chloroplast are permanently exchanging together, but in terms of redox, they are different. So that's you need to isolate. The cell needs to isolate some processes to maybe increase rates. So that's, to me, it's really important.
Yeah, and it's a different. I mean, it's a bit like what you were saying, that it's with the minute you're quantitative, you change your view on your system. So there is another question from Dimitri. Based on the available data, would one estimate volume of cytoplasm of different cell types? Example, bundle shape cells. Thank you. Have you, or is it possible to calculate the volume of membranes in different cell types? Okay,
So, yeah, so for the bundle shift cells, unfortunately, these cells have not been characterized a lot. We know for the chloroplasts, there's only maybe one study actually. I hope that with the new, it's not FIP-SEM, it's SBF, SEM, that's it, serial block phase SEM. We might go a little bit deeper in the analysis of these cells. But still it has to be done. So we don't know. They are very different. The vacuole is quite big. The chloroplasts are much smaller. So as yet, no. But it's something that is once we know that we need them, this kind of data, that's quite easy to do. We just need money and time and people to reconstruct the 3D structure.
Right.
Is it possible to calculate the volume of membrane in different cell types? So this is what we have in it's hidden in the table S15, and I realize that it's not finished actually. Then there still needs some calculation to be done, but you already have some information on that. You will need a hypothesis on the thickness of membranes, but it's possible. So you can calculate the amount of lipids in a leaf, and then if you're interested in lipid synthesis, that's a way to know how many lipids are involved in structure, how many lipids are involved in storage energy. So that's some calculation that could be done and could be injected in models.
Sounds good. I have another question. It's not a critique. It's more like to question the question. When you make an atlas, you have, as you said, a reference, and as you said, it's not perfect because it's a reference with certain conditions, etc. How do you deal with the risk of making it too stiff of a reference? Meaning that, let's say, if everyone goes with these values, and then we stop questioning that these values are, correct or not, or how do you go about keeping some flexibility, let's say, in a reference? It's a bit conceptual, I guess.
Well, this could be improved. This could be made visual somehow, because we can consider this as still a draft. And for example, in the values I have, we could add a column to include, for example, this variability of the rosette leaf area, even in well-defined conditions. We know exactly it's leaf 5, it's 6, etc., but there are some variability. That's something we could add. Maybe I could write this. So that indeed it's not taken as too stiff. That's a risk. After that, the other risk is that people generally take the reference value that goes in the good direction. For example, for chloroplast, if you want to say that it's the physiological range, you use the 30 microliter per chloroplast, 30 femtoliter per chloroplast, when the real value is 90. The 30 comes from old values when you isolate chloroplast with a high pressure, whatever. So the risk is also there to pick up, to do some cherry picking. But it's true that, well, that's a risk that needs to be evaluated and maybe corrected by this providing range, not thinking in terms of precise value, but range envelope of probable values.
I have to give room, but it's in the table already, and to some extent, so it's already. Maybe I don't know if people have other questions. Maybe maybe one last question from me. What's the next step? Of course, what do you want to do? I mean, this is a huge amount of work, and I can see that this is evolving as it is. But can you tell us a little bit what you think for the future?
So in the near future, we could have, as I said, another lab published a block of a leaf just to analyze the chloroplast volume. So actually this block is full of information, and they agree to send us the crude data. We have people here to reconstruct, so we will have much better data for veins, because by chance they grew the plants in long days, and it was at the same nearly the same stage. So this is fine. There is something that I have in store. This is about fluxes. I also collated many values of fluxes from many different studies. So we could do the same with metabolite. For steady state flux analysis, it could be useful. And the dream would be the whole plant atlas. So that's a question of money, of time, of it's. I think it's quite difficult to convince people that we need money to simply do cartography of a plant. I would choose a very simple plant, the seedlings at the cotyledon stage, because you already have all the cell types. Some work has already been done on cotyledon in Switzerland. There's the paper by Rosa Pipiton. We used some data from these chloroplasts, because chloroplasts in the cotyledon are similar to chloroplasts in a leaf. Then that would be, we would have all the cells, even in the roots. And this plant stage is very interesting, because you can grow it in, we have the device here, you can grow it in bulbic water, and then, or Evian water, and they are blocked. They are very low nutrient amount, they're blocked, but then you can add what you want and you can start the development. So that's, to me, it's a very nice stage, quite simple, not a lot of cells. It would be the Cenorhabditis elegans of plants.
Right.
And then we can do many different things. I just need to convince people that we need money to do that.
Oh, sure. Yeah, no, definitely worth it. Yeah, hey, super interesting. There's another question from Olivier Martin. Is there a reason for the vein cells to be so small in a similar vein? Playing with words, what is the fraction vacuole volume over cell volume in epidermal cells?
So in epidermal cells, it's huge. It's 99% of the vacuole is the epidermal cells. It's huge. I think this is simply because the epidermal cells and the function that is optimized. Sorry for the term. It's probably to store flavonoids or molecules or defense molecules. You need to have a lot of in there. It has to be transparent. It has some specific properties with light to diffract light. So that's huge. Is there a reason for the vein cells to be so small? Well, their function is not to pick up light. Their function is to transport. So the smaller they are, the higher the concentration would be. So the higher the rates would be. I made a funny calculation. If you look at the volume of veins in the leaf, compared to the total volume of leaf, it's similar now if you consider surface to the roads in UK. What are the percentage of roads in UK? And I heard recently that people in UK do not want any more new roads. And maybe because there are behind that some optimization principle. A well-connected country, it has a certain amount of matter invested in the transport, and you still need place to fix light to capture CO2. And this is a question of surface. So that could be an explanation. And it also costs a lot of money, carbon, because xylem cells must be thick. So the thicker it is, the more solid they are, the plant will resist to drought. And there are also some capillarity issues. The smaller it is, the less sensitive to cavitation, for example. So there might be some physical principle behind that could be explained.
Yeah, I agree actually on so it was not for vein cells, but we showed this for epidermal cells, right? That if you reduce, we work with Richard Smith, if you reduce the diameter, you reduce the stress level in the hole as well. So I mean, this could go for capillarity as well. Yeah, I agree. Cool. Thank you very much. If there is no more questions,
Yeah, there was another question. Would you consider making a similar atlas for other model plant species? Well, I think it could be great. The difficult part was to build this Excel table. If now we have the data, 3D data for another plant, that would be quite a little bit, well, it would take time. It could be a little bit boring, but it's feasible because we know where to place the values. The equations are written in the table, so that could be done. And if someone wants to do it, please take it and do it. That's make it your own and make this evolving. It's also an answer to your question of something that is too big to fix, too fixed. Well, take it, make it evolving, and I would be happy with that. There's no problem.
That sounds good. But it's true that you also have an experience, right, with building an atlas. So that's definitely something that you can share also,
Yep.
with the people. Thank you very much. This was enlightening.
Thank you.
And, also helps for the readers to have a way to go through the paper and to find the resources that are really super important for anyone interested in development and morphometry and quantitative plant biology.
Okay.
Casey, the floor is yours.
Perfect. All right. Well, if there are no other questions, I hope all of the questions were answered and you all enjoyed this webinar today. I really want to thank Dimitri, Gilles, Olivier, thank you so much for taking time and showing us all this information today. And thank you so much for all the attendees. Thank you for the questions. We really appreciate you attending. If you're interested in more of these kind of talks, go follow us on X, Twitter, whatever it's called these days, at QPP_journal. And we will be posting this talk later as well. If you missed any part of it, you can watch the recording later. But thank you all again so much, and we'll see you at the next one.
Thank you. Bye.
Everyone.
Bye.